Greetings from EMBL in Grenoble, where it's 31 degrees. It's summer before its time. I saw on the list that there were people alumni from Grenoble, and so many greetings to Valerie, I can see, and Ru Yan, who's in Shanghai, if she's there, and of course Andrew Miller, who is the first head of EMBL Grenoble. In fact, I joined the lab when he was head now many, many decades ago. So I've been here an awful long time, but I'm still here working on influenza virus mainly. So what I'm going to talk about to you today is, can we repurpose any anti-influenza drugs for use against the coronavirus? So can you go back? Yeah. So the next one. Yeah, this is what I'm going to kind of talk about quickly. I hope So influenza and SARS are pretty similar in some respects, but basically their molecular biology is very different. They are both envelope viruses. They both infect the respiratory tract, and in their severest form, they can both cause viral pneumonia and hyperinflammation of the lungs. Both are easily transmissible via droplets. Both have proven pandemic potential and potentially kill hundreds of thousands, if not millions, around the world. However, the molecular biology of the two viruses is completely different, except for the fact, and this is what I'm going to focus on, that they're both single-stranded RNA viruses, and therefore both possess an evolutionarily conserved RNA-dependent RNA polymerase that replicates the viral genome. And you heard a bit about that from Peter. Even so, they are quite different families of RNA viruses. Influenza virus is a negative sense virus, which means its genome is, and it's also segmented in 8 pieces. So the virus contains 8 pieces of negative sense genomic RNA, which means the viral polymerase has to first transcribe the genome before viral messenger RNA is available. Furthermore, influenza replicates in the infected cell nucleus. Coronaviruses have a positive strand RNA genome that you heard is unusually long. It's about more than twice the coding capacity of influenza. And coronaviruses replicate in the cytoplasm, and the positive sense means that genome can directly act as messenger RNA without being transcribed first. So my group has been studying influenza RNA-dependent RNA polymerase for the last 15 years or so. And this polymerase has many unique features, and our goal has been able to understand it mechanistically by structural biology. Our latest results using cryo-electron microscopy, where we were able to follow the complete cycle of transcription, were just recently published in Cell. In parallel, we've been using, and others, our structural information which has been underpinning development of the next generation of anti-influenza drugs that target directly the influenza polymerase. So let me just remind you in the next slide what a polymerase does. So the circle here represents the polymerase. The yellow is a sequence representing the viral genomic RNA, which we call the template. This binds into the active site of the polymerase, and then also binds what's called a primer, a short RNA sequence, which anneals to the end of the template. And then there's a binding site for an incoming NTP, in this case ATP, which is selected because it's complementary to the U in the active site. And then the next slide, skipping a few steps, what happens is that the template and the product subsequently translocate one nucleotide at a time as each incoming NTP is added to the product in a way that's complementary to the template. So like this, the polymerase chugs along the whole of the template, making a complementary copy of the other sense strand. So the question is, with influenza virus, has a very special mechanism for deriving this primer, the part that's in blue. Well, first of all, this was the structure of the polymerase that we determined about 6 years ago now. And here you see this yellow template end of the RNA going into the active site. One of the curiosities about flu is that it actually binds the complete other end of the genomic RNA as well, what's in pink there, and that activates the polymerase. But on top are shown these two extra domains called the cap binding domain and the endonuclease. The next slide. These are used in snatching capped RNAs from the host cell polymerase II. I remind you, this is happening in the nucleus of the infected cell. The viral polymerase binds to the host pol2, which is churning out capped RNA transcripts. And these are bound by the cap binding domain of the viral polymerase and cleaved by its endonuclease, giving this red capped primer, which is then rotated into the active site to initiate transcription, as I showed you in the previous slide. So this is an incredibly special mechanism, but when we solved the structure of these domains, we could see that they would be ideal targets for antiviral drugs. So without going into a lot of details, over the last 10 years, 2 new classes of anti-influenza drugs have been developed. One is shown on the left here, exemplified by Remdesivir, developed by Vertex and Janssen, and this is a cap analogue. So it binds and blocks the cap binding domain of the viral polymerase and stops it doing any cap snatching and therefore stops transcription. An endonuclease inhibitor, so this is the other domain which cleaves the primer off from the nascent transcript. And here we have Remdesivir, which is now marketed, actually. It's been on clinical use in USA and Japan since 1 year now. It's marketed under the name of Xofluza, and this potently blocks the endonuclease of the influenza virus. So these 2 drugs are real breakthroughs, if you like, in the influenza field. I'm telling you all this only to say that they're completely useless against coronaviruses, because this is an incredibly specific mechanism of influenza virus. And in fact, coronaviruses don't need to cap snatch anything because they have their own capping enzymes encoded in the genome. So now let's focus in more closely. Here's a comparison of the structure of influenza polymerase, which we determined with the primer template RNA, and a incredibly recent structure just from a month ago of the SARS-CoV-2 polymerase, again with a primer template RNA. You can see they're different in size, shape, and everything when you look at it like this, but you'll see that the RNA, the red and the yellow strands are pretty much the same. And if we focus a bit closer on the next slide, you can see that the core of this enzyme is virtually identical. And that's because they're doing this very evolutionarily conserved mechanism of nucleotide addition, template-directed nucleotide addition. So this is the real core of the dependent RNA polymerase. So the question is, can you target this highly conserved region? And the answer is yes. So here is a drug that was developed initially against influenza. It's called favipiravir, or T705. And this is a class of drug called nucleoside analogues, which mimic ATP, GTP, CTP, or UTP and bind indirectly in the active site of the polymerase. And this particular one mimics actually ATP and GTP, and it functions what's called by lethal mutagenesis, which means that once it gets incorporated into the product strand, you're losing coding fidelity because the included nucleotide analog can mimic either an A and G and base pair with a U or C. So any subsequent copying of the genome strand will give rise to errors. Similarly, the next compound does the same kind of thing. It's a cytidine-uridine analog, which again causes lethal mutagenesis. Now, the first one was approved for use against pandemic influenza by the Japanese government and is now being resuscitated as an anti-SARS-CoV-2 drug. And as with all these new drugs, there are several that are being tried out. Some clinical trials are showing some effect, but overall, we've not got enough data to really claim any of them are working particularly well. The one at the bottom is not even in clinical development, but it works in vitro. Again, this was published very recently in Science Translational Medicine. And all this brings me to Remdesivir, because this is in the same class, and this is the one that everyone's talking about now. This was developed by Gilead to inhibit Ebola polymerase. And an Ebola virus is an RNA virus as well. It's a negative strand virus, but not quite the same family as influenza. Anyway, this drug again works by directly being incorporated into the product by the RNA polymerase. And then this works in a slightly different way. It's thought to be by what's called delayed chain termination. In other words, it is an ATP analogue that's incorporated opposite U, but it doesn't actually stop the polymerase until 2 or 3 more nucleotides have been incorporated. And then for steric reasons, it just seems to block translocation. Anyway, this, as I'm sure many of you are aware, has been in clinical trials. So there was some kind of positive results from a clinical trial in the USA recently showing it shortened the time it takes to recover from COVID-19 by a few days, and hence has been authorized for emergency use in the US and Japan. And one of the issues with this is there are side effects. It's a bit toxic. In fact, all these nucleotide analogues tend to be because they can interfere with other polymerases, cellular polymerases. But this one is also not generally in clinical use up till now, and therefore its availability is very low. It's an intravenous drug as well. And I read recently that Gilead was hoping they might have 1 million doses made by the end of the year. So with that, I'm going to finish again with a summary. So I've gone through all this. The recent flu drugs are very good against flu, but no use against COV-2. But these other nucleoside analog drugs may be useful. They're more broad spectrum, but rigorous clinical trials are really needed to see how well they'll perform. So with that, I'll stop, and if there are any questions.
Thank you very much, Stephen. That's fantastic. I notice a couple of things. One thing, my understanding from the recent media reports is that Remdesivir shortened the duration of the illness for patients who recovered anyway, but didn't actually reduce the mortality rate or prevent the number of people who died. Is that correct?
That is correct for that clinical trial. Shortened from 14 to 11 days, something like that, which could be significant. But obviously none of these are a magic bullet yet, and I don't think any of the drugs available are going to be magic bullets, and we're just looking for something that helps a bit, because to get a new drug from zero on a drawing board is usually 10 years. And
I noticed you didn't talk about the clinical trial being carried out by President Trump on chloroquine on himself.
Yeah, what can one say?